Matthews, and you are about to watch my biology video, which of course you already know. So I am here at the WashU Medical Campus downtown, and my project consists of working with the Galbert Lab over in the McDonald's Science Building and examining their magnetic tweezers experiments, which I will give you more detail as we go through the video. So up next, I will be showing you the magnetic tweezers device. So stay tuned. So here we have some data from the magnetic tweezer device, and this is a magnetic bead with one strand of DNA attached binded to it. And so even though it looks pretty big, and you probably can't imagine this, it's actually 1/1,000,000th of a meter, or a micrometer. So, and if you look to the left, you will see the magnetic tweezer device. And so this device is actually a microscope, as if you hadn't gotten that from before. This part of the microscope sheds light onto the slide, and this down here is the neodymium magnets, which can move up and down, as pictured, videoed, or they can rotate around. And scientists use this quantitative data to study biochemical and biomolecular processes in the DNA, which we'll get more into in a minute. What I'm currently showing you is a video of magnetic beads binding to a single strand of DNA per bead. The reason that DNA and magnetic beads bind together is because of two special molecules that bind together called streptavidin, a protein, and biotin, also known as vitamin H. The process of getting to what you see now starts by flowing magnetic beads coated in streptavidin into the magnetic tweezers. After the DNA that has been altered to have biotin in it, in a process called DNA synthesis, is then flowed under the magnetic tweezers and immediately binds to a streptavidin-coated bead because the two are attracted to each other. The reason you know that there's DNA in the beads is because of a first experiment where you change the control variable to water and flow the beads and DNA under. Since the beads don't show up with the water but do with the streptavidin, then you know there's DNA bound to the beads. Alright, so now, unfortunately, I've gotten to the end of my project where I'm going to present my conclusion, and then Professor Eric Galbert is going to give the future directions of the lab. But in conclusion, I have learned a lot through the processes of the magnetic tweezers, and now I'm finally going to tell you how it's going to be important to the world. So basically, through this research, they can help with gene regulation, which regulates cell behavior and what's going to do what, and how it can affect what you look like, and internally, externally, and stuff like that. So that's been very interesting, and I didn't know that things on such a small level could affect us in such a big way. So that was really something to do, to learn, and I'd really like to thank the Galbert Lab for this. So, go.
Okay, so hi, my name is Eric Galbert. You haven't seen me yet to the very end of the presentation, but that's okay. First, I just want to thank Chris for contacting me. It's been a lot of fun interacting with him and with James as well, and learning what they're discussing in class, and hopefully teaching them some new things. So future directions of the lab are largely related to what we've already been doing, but we're really interested in understanding how transcription initiation occurs on a molecular level in bacteria such as Mycobacterium tuberculosis, which of course is a huge problem all over the world. So we're going to continue using our biophysical techniques like single molecule magnetic tweezers, single molecule optical tweezers, as well as more traditional biochemical techniques to try to understand these processes in the hope of eventually one day figuring out a way to help combat these bacteria that cause problems for us humans. And with that, I would just close and again thank Chris, and hopefully I'll get a chance to see some other scientists to be sometime in the future. So